Good afternoon, one and all. Myself, Dr. Mrs. Damini Motwani, Assistant Professor, Department of Biochemistry, Dada Ramchand Bakrus, Sindhu Mahavidyalaya, Panchpavli, Nagpur. Today I am here to present on the topic agricultural waste treatment using cellulase enzyme isolated from Aspergillus species. Introduction. An enzyme is a protein molecule that is a biological catalyst with three characteristics. First, the basic function of an enzyme is to increase the rate of reaction. Second, most enzymes act specifically with only one reactant to produce a product. The third and the most remarkable characteristic is that enzymes are regulated from state of low activation to high activity and vice versa. Cellulose is an organic compound, a polysaccharide consisting of linear chain of several hundreds to over 10,000 D-glucose unit linked by beta-1,4 glycosidic bond. This is the figure of cellulose. Cellulases are produced by various sources like fungi, plants, bacteria, etc. Now, the major reasons for increasing the interest in microbial sources. The production of cellulase is an economical bulk production activity. The microbes are manipulated to obtain enzyme of desired characteristic. The growth of microorganism is rapid and this will in turn speed up the production of enzyme. Microorganisms are easy to handle when compared to animals and plant. The microbial cellulases meet industrial demand. A large number of them are available commercially. Now the production methods. The production methods of any enzyme are of two classes, that is submerged fermentation and solid state fermentation, and it has been proved that solid state fermentation gives better result as compared to submerged fermentation. The solid state fermentation is a method used for microbes which require less moisture content for their growth. The solid substrate commonly used in this method are bran, bagaces, paper pulp, etc. The main advantage is that nutrient-rich waste material can be easily recycled and used as substrate. Now, there are certain advantages of SSF, that is solid state fermentation, over submerged fermentation. Solid state fermentation, simpler equipment, higher concentration of product and lesser effluent generation. For several such reasons, SSF is considered as promising method for the commercial production of enzymes. There are various applications of enzymes in paper pulp industries, detergent industries, textile industries, foods and beverages, tomato, ice creams, etc. Now the methods and material used in the study. Collection of soil samples. Soil sample was collected from food market in a sterile condition to avoid contamination and transferred to laboratory using sterile polythene bags. Isolation of fungal isolate. A suspension of fungal isolate was isolated. A suspension of soil sample and sterile distilled water was prepared and plated on potato dextrose agar. Isolation of cellulitic fungal isolate. The cellulitic fungal isolates were screened by growing on starch agar medium. The fungus Aspergillus niger gave maximum clear zone and was further selected as potential strain for cellulase production. Extraction of cellulase. The fermented medium was centrifuged and cell-free supernatant was used for estimation of cellulase activity. The cellulase activity was measured by Miller's method. The absorbance was measured at 540 nanometer by photometric colorimeter. A standard graph for cellulase enzyme was prepared. 1 unit of cellulase activity was described as an amount of enzyme that released 1 micromole of reducing sugar per minute under standard conditions. Now the results. Effect of incubation period on cellulase production. Optimization of incubation period is an essential parameter for maximum growth of Aspergillus niger and hence generally it affects cellulase activity. The results as shown in the figure reveals that incubation period, that is 6th day, was found to give the best cellulase activity. This figure can clearly shows that the 6th day gives maximum activity. Effect of temperature on activity. Incubation of fermentation medium at various temperature was performed. The figure shows that maximum cellulase activity was observed at 40 degrees centigrade. You can see in the figure that 40 degrees centigrade gave maximum cellulase activity. Effect of pH. In the effect of pH, you can see that the pH 8 is giving the maximum cellulase activity. Effect of agro waste. From the agro waste, it can be seen that the wheat bran is the agro waste which is giving a much cellulase activity. The conclusion: nowadays enzymes are used for various reactions because they are biodegradable and can produce using biological sources. The fungal strain Aspergillus niger were isolated and screened for cellulase production by a clear zone formation. The results suggested that fungal isolate gave maximum activity. Its potential strain can be easily degrade cellulose. The effect of various process parameters of cellulase activity was found to be influenced by incubation period, temperature, and much more parameters. These are my references. Thank you.